Even more traffic this morning. Good morning. Good morning. Awesome. Hi. I'm going to have to send out another email reminder. I just sent out an email reminder on attendance. I'll have to send out another one reminding people to try to show up on time. Not easy. I don't know why. Not easy in an online format. I know it's not. But we will get you here. All right. Well, we got a lot to get through today, and I'm going to go ahead and get started because it is class time. So I just want to remind you that we've gone through quite a bit at this point. That should be obvious, but it all ties together. So we got cardiac output. We know cardiac output is the function of the heart, and that there are two primary variables that affect cardiac output. We got heart rate, and we got stroke volume. Heart rate is determined by autorhythmic cells. So if we were to write down the two types of cardiomyocytes, this is one way to go about doing it. We know that one, there are autorhythmic cells, and two, there are contractile cardiomyocytes, or just myocardium, we'll call them that, myocardium. The autorhythmic cells, well, man, those are controlling the heart rate. These guys about the heart rate. Lovely. Contractile myocardium, they control the forces inside that heart. Okay, they control the force of contraction, but that force of contraction is controlled by, what did we learn yesterday? Force of contraction is proportional to what? Good, calcium. So we got this contractile myocardium. It's going to be all about calcium, and that calcium is going to drive the force of contraction. But it's not just about the force of contraction, it's about the blood that's inside those chambers, and that's something we're going to focus on in today's lesson. But as that muscle contracts, it's going to put pressure on that blood underneath, and that blood is going to go shooting out of the heart, and that is stroke volume. So the force of contraction is going to definitely help determine the stroke volume. We're going to see there's some other variables that can affect stroke volume as well. But that's the job of the contractile myocardium, is they help control how much blood leaves the heart in each heartbeat. So cardiac equals heart rate times stroke volume. Sweet. Autorhythmic cells, heart rate, contractile myocardium, stroke volume. We have a functionality of each one of those that applies to the total heart function. Lovely. It's what we want. Now, for each one of these, we've been learning about action potentials. There's an action potential for an autorhythmic cell. There's an action potential for a contractile myocardium. You guys should be able to draw that for your exam next weekend. And not just draw it, but understand each part of it. You're going to need to tell them exactly when the gates are open, exactly when the gates are closed. And you're going to need to tell me which ions are crossing the membrane and in which direction. Shouldn't be too crazy, though. We've done it many times, right? For both the autorhythmic cells and the contractile myocardium, we have lots of examples of those action potentials. So I hope you guys can go and study those. Draw it in BB. You'll need to draw it, and then you'll end up, if it's on an exam, then you'll upload it as you just draw it on a piece of paper. I'm sorry, I'm so sorry, I'm not understanding. You'll just draw it on a piece of paper, then you upload that to as a PDF onto your. It's not very difficult to do. So I'll always make you guys do something like that, a flow chart, something along those lines. You'll just draw it on a piece of paper, take a pic, upload it. Yeah, it's It's the easiest way to learn. It's also the easiest way to put down your knowledge. It's just with diagrams, flowcharts, graphs, that sort of stuff. Cool. Now, that is not where we stopped, of course. Where we stopped is during this idea of, can a patient change their cardiac output? If they need more blood, can they change their cardiac output so they get more blood? And the answer is yes, right? Can we increase our heart rate? Of course we can. And if we increase our heart rate, that will result in an increase in cardiac output. Can we increase our stroke volume? Of course we can. And if we increase our stroke volume, that will result in an increase in cardiac output. Assuming the other one doesn't decrease. You can increase heart rate all you want, but if there's no stroke volume, cardiac output is still zero. What's infinity times zero? Still zero, because anything times zero is zero. Does that make sense to everybody? All right, and we are walking our way through how these are changing, and we focused only on heart rate so far. So how can we change heart rate? Okay, let's draw this out in a slightly different way. Yeah, directly proportional. There's all sorts of different ways to study. That's what I'm trying to teach you guys. Find one that works for you. But cardiac output equals heart rate times stroke volume. Well, guess what? We can do this as a flow chart. So let's do that. Cardiac output equals what two variables? Heart rate and stroke volume. Can we change heart rate? Sure we can. What will have a positive actually, let's start with a negative influence, because positive we want to tie into this other one. Let's start with what can have a negative influence on heart rate. What's going to decrease heart rate? That's what I mean by negative. Exactly. So down here I'm going to write parasympathetic nervous system, right? So the parasympathetic nervous system will, there's a little negative sign, decrease the heart rate, and therefore, if we decrease the heart rate, cardiac output would decrease. So another way to put lots of information in one flowchart. What else can we write in here? What do we know about parasympathetic nervous system? What neurotransmitter is released for the parasympathetic nervous system? Always on the target organ. Acetylcholine. And since this is heart rate, what type of cell do we know the acetylcholine is being dropped on? Which cell is this being dropped on? So if you don't know that immediately, then as you're studying, you can write in, heart rate, this is determined by AR. You could add that to your flow chart just in like a subset. You don't want too much information. What else do we know about acetylcholine? What receptor on the target organ are we always dealing with acetylcholine in the parasympathetic nervous system? On the target organ, Andy, on the target organ. Nicotinic is correct if we're talking about skeletal muscle, but that's the only target organ in your body that has nicotinic receptors. There are no other target organs in the body that have nicotinic receptors. Well, not really, but we're not going to deal with those other ones. I'd love more people to write muscarinic, just so that you guys get that little link in your brains. On a target organ, that means on the target tissue. That means on the heart, on the autorhythmic cells, there are no nicotinic receptors ever. No. Nicotinic are upstream from that, right? They're at that pre and post ganglionic synapse. No, here it's always muscarinic. So on autorhythmic cells, we have muscarinic. You guys didn't learn this yesterday, but I want to make sure I have enough room. Muscarinic is written out as a mu sign. I don't know if you guys know mu, but mu is this Greek M letter. Kind of looks like a cursive M. we got receptors, muscarinic receptors on the autorhythmic cells, and they help control heart rate. Cool, huh? Just a nice way to summarize what we're doing. Now, I don't know, I'm just going to change color because I like to change colors, but can I also impact the heart rate in a positive way? Can I increase heart rate? Sure, I can. So what's going to do an increase in heart rate? Sympathetic nervous system. Now here's a trick question. What neurotransmitter is always released onto the target organ from the sympathetic nervous system? It's a trick question just because it's tricky. It's not a trick question though. Never acetylcholine. Acetylcholine is in the parasympathetic nervous system. Remember, we're talking about the target organ. I am talking about the actual autorhythmic cells. You guys keep trying to go back to the acetylcholine, but that's at that pre and postsynaptic junction. So that's why this is a trick question, okay? Because Heather, Parker, Andy, I love that you're writing epi and norepinephrine, but epinephrine is never released from a synapse. Epinephrine is only released from the adrenal medulla into the blood. So epinephrine is always a neurohormone, never a neurotransmitter. Does that make sense? Neurotransmitters are always released at a synapse where there's a synaptic cleft, and it's going to directly synapse with the target organ. Does not get into the bloodstream. Does that make sense to everybody? So if I have an innervation, if I have a neurotransmitter, I'm talking the neurotransmitter, not neurohormone, that's impacting the heart autorhythmic for the sympathetic nervous system, it can only be norepinephrine, never epinephrine. That is not a neurotransmitter. But if we have an emergency response, then the adrenal medulla is going to be activated, at which time a combination of norepinephrine and epinephrine get out in the bloodstream, and these would be considered neurohormones. Does that make sense? This here is only emergency, though. Emergency only. I see you, Heather. 1 second. But does this make sense? We need the sympathetic nervous system if you just stand up out of your chair because you need to get more blood up to your brain because you just stood up. Are we going to have epinephrine dumped in the system if you stand up out of your chair? No, that's not an emergency. We just have to release a little bit of norepinephrine out onto that heart. So we do that via just a normal synapse. Yes, no, maybe so? Heather, how can I help?
So I'm confused by what you said about that. You said that those are, norepinephrine, epinephrine can only be released from the medulla, not through the nerves. But then you said norepinephrine can.
I said epinephrine. I'm sorry, I miscommunicated then. I'm not sure what I said before because I never remember what I say after the fact. But epinephrine is only a neurohormone. And when norepinephrine is released from the adrenal medulla, it's a neurohormone. But when norepinephrine is released from a synapse, from a terminal bouton at a synaptic cleft, and is released directly on a target organ, it's called a neurotransmitter. Does that make sense?
Only norepinephrine can do that, not epinephrine.
Epinephrine is never released directly at a synapse onto a target organ. Epinephrine is only released as a neurohormone into the bloodstream from the adrenal medulla.
Okay, thank you.
You're welcome. All right, is everybody on board with this? And then, of course, what receptor is it binding to on the heart? And let's be as specific as possible. What receptor are we dealing with here? Adrenergic. Good, Veronica, but what kind of adrenergic? Yesterday we spent a lot of time on 5 different types of adrenergic receptors, and only one of those is found on autorhythmic cells. Yes, it's beta 1. Good. That's going to take you guys a little bit more practice. Practice? This is beta 1. I should have given myself more room. Adrenergic receptors. Cool. Just as a quick word, if I am going to be changing the heart rate with any drug whatsoever, but norepinephrine, epinephrine, acetylcholine, I will be changing the heart rate. What type of am I? What type of drug am I? Good, Andy. Nope, no. I'm asking for this general broad-based word that Andy just wrote down. Do you guys remember this word chronotrope that I talked about yesterday? Chronos, like chronology. Chrono means time in Latin. So that's, you know, if you're putting things in chronological order, you're putting them in an order of time. So chronotrope tells you that we have something that's acting on the heart rate timing. Everybody feel pretty comfortable with that idea? So anything that impacts the heart rate is called a chronotrope. If it has an increase in heart rate, it's a positive chronotrope. If it has a decrease in heart rate, it's a negative chronotrope. Okay, so we got flow charts. We got them coming out of our yin yang. We got stroke volume we still need to work on. But what I want to do is I want to write another flow chart that's almost this exact same thing right here, but we're just learning it in a different way. We're going to focus on the sympathetic nervous system. And we're going to say, let's activate the sympathetic nervous system. But here's the deal with activating the sympathetic nervous system that you guys haven't learned until I'm going to teach you now. Is that there's a portion of the brain that is primary controller of the sympathetic nervous system when it's controlling the heart. So anything that controls the heart is coming from a portion of your medulla oblongata back here called the cardiovascular control center. The cardiovascular control center. You got it. You've been reading ahead probably, I hope. Therefore, Becca, you nailed it. Okay, this is the cardiovascular control center. The cardiovascular control center has, well, two major parts on it, as you might be able to tell from the name. Yep, lab quiz did. The lab quiz that's not due yet, but yeah, the lab quiz from yesterday's lab, right? Oh, actually the one before that? Baroreceptors. Yeah, that's right. So good, you guys already have. Yep, that means you guys already have a background in this, and that's fantastic. So cardiovascular control center has two major parts of it. One is a cardio, and the other one is a vascular, right? So we got a cardiac control, and we have a vascular or vasomotor. Perfect. Vaso tells us vascular, motor tells us that we're working on muscles related to blood vessels, which means vasoconstriction and vasodilation. We'll end up talking a lot more about these vasomotor inputs later, some a little bit now. Cardiac control. Well, cardiac control can either be excitatory or inhibitory. So we have a cardio acceleratory center, and we have a cardio inhibitory center. Coming together, kind of. We have a way to speed up the heart, and we have a way to slow down the heart. Well, what part of our nervous system do you think the cardio acceleratory center controls? I'm going to speed up the, oh yeah, you got it. You got it, right? We got the sympathetic nervous system right here. And then what do you think about the cardioinhibitory center here? We know this is going to be controlling the PNS. So golden, golden, golden. So cardioacceleratory is going to, boom, it's going to hit the sympathetic nervous system, sympathetic trunk ganglion. Yes, right here at synapse, acetylcholine is released on nicotinic. But about the target organ for the sympathetic nervous system will always release norepinephrine as the neurotransmitter onto the heart, and it's going to act on beta-1 adrenergic receptors. Yes, no, maybe so? We're going to go ahead and write in this abbreviation CVCC because for this class, the abbreviation is good enough for me. You guys, I mean, I highly recommend you learn what CVCC stands for, Cardiovascular Control Center, because it tells you what its function is. But on a test, you can just write in CVCC. So as an example of that, we're going to say, let's activate a portion of the CVCC. Okay, and this portion of the CVCC that we're going to activate is going to be cardioacceleratory. So we're going to go cardioacceleratory portion. Exactly, you got it. So CVCC, cardioacceleratory. Now, what happens next? Well, if we want to draw it as a picture, right, basically you'd end up with this picture of a neuron that starts way up in that medulla oblongata. It's going to come down the spinal cord. It's going to synapse in the spinal cord. It's going to go out into the sympathetic trunk ganglia. It's going to synapse there, and then it's going to send a big, long axon down to the heart, and then it's going to act on the heart from there. Right? That would just be a simple diagram of that function. Well, we can do that with a flow chart. CVCC, cardioacceleratory, but let's skip all the middle stuff because that was supposed to be tested on in A&P1. So CVC, cardioacceleratory center, let's go ahead and kind of just say, well, sure, there's some nicotine and stuff in here, and that's going to be a cool thing for us to talk about, but not essential in the function of the heart. So let's skip it, and we're going to get down to the neurotransmitter that's secreted on the heart. So if we activate the CVC, and we're going cardioacceleratory, one last time, what neurotransmitter is going to be released onto the heart? Norepinephrine. Beautiful. And now, what receptor is going to be activated by this? Beta-1 adrenergic. So we're going to A with an arrow up, beta-1 adrenergic. But where did we find beta-1 adrenergic receptors? Do we find them just on autorhythmic cells, just on contractile myocardium, or both, everywhere? Where do we find them? We find them everywhere, absolutely everywhere. So let's focus on one of those targets right now, just as we're still kind of reviewing yesterday's material, and we're going to say we're on the autorhythmic cells. So norepinephrine is going to be released onto autorhythmic cells, which is a nice little descriptor because what does that tell us it's impacting? What part of the cardiac output equation do autorhythmic cells help us control? Rate only. You guys with me? So we're going to be dealing with the heart right here. So let's continue. Beta-1 adrenergic receptors. What type of receptor are they? Are they chemically gated receptors? Are they receptor enzymes? Or are they GPCRs? These are GPCRs. These are GPCRs. We went through this yesterday. This is the one at the very end of the lecture. That's why I'm going through it again. But this is the one that had this idea of G proteins being stimulatory or G proteins being inhibitory, and beta 1 adrenergic receptors, autorhythmic, norepinephrine, cardioexcitatory. So we know this is going to be increased in the heart rate. So this is where a G stimulatory protein is going to come into play. If we're going to increase the heart rate, what do we want to do to the rate of polarization? Increase, decrease, no change? Good. Good. So we're going to activate a GPCR, and somewhere down the road, we're going to increase the rate of depolarization. So what two types of transient receptors or transient transporters, sorry, not receptors, transporters, are we going to be dealing with here? Perfect. Funny and T-type calcium channels is the exact answer, but sodium and calcium will get you there, Tariq, so you're not wrong. So we're going to activate some beta-1 adrenergic receptors. Somehow we're going to impact those funny and those T-type calcium channels. So let's fill in the middle. That's the hard part. When we activate the G protein, that G protein is going to go and activate an enzymatic protein found on the cell membrane. What's the name of that enzymatic protein? It's called adenylate cyclase. Don't be an oops, you're all right. I like what you did. You actually were talking about how GTP displaces GDP. So I skipped a step and you didn't. So that's awesome. Good job, Andy. Right? But this GPCR is going to go activate adenylate cyclase. And adenylate cyclase does this hydrolyzation step where it hydrolyzes ATP twice. So it's going to lose 2 phosphate groups. So it's going to take an ATP and it's going to go and give it a chemical reaction, little twist, little bang, little pow, little magic. And what are we going to be left with? Cyclic AMP. I always test on cyclic AMP. You got to understand this little step right here. I love cyclic AMP because that's what caffeine is impacting. Okay? I love cyclic AMP. It's my favorite because I'm addicted. So now this cyclic AMP is going to go and act on some other proteins inside the cell. It's a secondary messenger, so it's really powerful. It does all sorts of stuff, but one of the primary things that it's going to do is it's going to go act on kinases, protein kinases, PKs, and there's PKA and PKB and PKC and PKD. You guys get the point. There's lots of PKs. But protein kinases, well, a kinase is an ACE, A-S-E, ACE. And all kinases have the same function, kinase. ACE is an enzyme, so perfect on that. What a kinase does, whether it's protein kinase A, B, C, D, there is no E, but whatever. Kinases do a process called phosphorylation. And I use this word on my exams, phosphorylation. Phosphorylation. I use this word on my exams and my homework questions and stuff. So you might come across it. And you'll need to know what the heck's going on with this word. There's called phosphorylation and there's something called dephosphorylation. All right, let's find out what phosphorylation is first. Here's the root of the word, phospho, right? What ion is critical in the body function that has to do with a phospho? It's a phosphate, right? So phospho has to do with a phosphate, like ATP is adenosine triphosphate. Your bones are made out of calcium phosphate composition. Your bones store away so much calcium and so much phosphate because the body needs so much calcium and phosphate. Does that make sense? Phosphates are all over the place. And they're normally floating around as an inorganic. They don't have carbon, they don't have oxygen. They're just this, phosphate group with a whole bunch of charges around it. So we're going to go ahead and just write that as a P, a lowercase I, and then we put a circle around it, and that's called an inorganic phosphate. So let's go with what we drew out yesterday. If I take an ATP and I hydrolyze this ATP, I'm going to be left with an ADP, right? I get rid of a phosphate, but what else is a byproduct of this? I lost a phosphate group from an ATP and ended up with an ADP, so now I get that inorganic phosphate as a little side group. Does that make sense? It's a byproduct of this hydrolyzation process. What else do we know about ATP? This is energy. This is energy, and it's actually, you got your adenosine, I'm just going to write it in as an A, that's bound to a phosphate, that's bound to a phosphate, that's bound to a phosphate, and the energy of all chemicals is stored in the chemical bond. So if I take ATP and I break that chemical bond, we're not going to worry about heat here, Tariq. You're not wrong. It will produce heat, but we're just thinking about the energy. So if I take this chemical bond that's storing that last phosphate, it's going to release energy. Does that make sense? Because the energy was stored here. It takes energy to make it a chemical bond. Therefore, it releases energy when we break that chemical bond. And that is going to be carried with that phosphate group. That energy gets carried with that phosphate group. So if we go through a process called phosphorylation, then what a kinase does is it hydrolyzes an ATP, releases an inorganic phosphate, then that inorganic phosphate can go bind to another protein, and when it transfers the energy to that protein. This is phosphorylation. When we move phosphate groups around, it's going to bind that phosphate group to this protein, and when it does, it provides energy to that protein. Does that make sense? Phosphorylation's moving phosphate groups around, providing energy where it needs to go. What would be a dephosphorylation? We cleave this phosphate group off of whatever it's bound to. So, pretty simple idea. So all pKa's phosphorylate things. That's what they do. All protein kinases phosphorylate things. So let's go one more step downstream. This pKa is going to act on funny channels, because now we're finally here, and T-type transients. And if I activate both of those, I'm opening them up a little bit more. I'm not really activating them because they're leak channels. They're always open. But I can make them open a little bit more or a little bit less, and pKa opens them up a little bit more. So what's going to happen to sodium influx? Increase, decrease, no change. And what's going to happen to calcium influx? Increase, decrease, no change. And the combination of those two things is going to increase the rate of depolarization. We hit threshold faster, which of course all leads back to heart rate, and that's going to increase that heart rate. How do you guys feel about this so far? Other than probably need to go practice it some more, but we've covered it a few times. This isn't crazy. I promise you, write it down, just a couple of times. And by the way, let me give you guys another study tip that I might have already given this semester, but I'm going to reevaluate, is that you should never, ever, 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 in my opinion, you should always practice, but you should never write this down on one piece of paper and then write it down on the same piece of paper somewhere else. You should never look at your previous notes as you're writing down your new notes. You should work your darndest to do it all from memory. And that's why I like whiteboards. You write this down, you erase it, and you write it down again. And if you make a mistake, you fix it. So you write it all down, you go back to your original notes, you say, oops, I missed that. Oops, I missed that part. You fix it, then you start from scratch and you do it again. And if you do that, it'll take maybe 5-10 minutes of your life, and it's going to stick as a memory better than if you just look at your notes and read this shit. Now, you want to make a professor mad, go that route. Just read through your notes and act like you know what the heck you're talking about. That doesn't do it. Nobody, unless you have this photographic memory, can do that. Start from scratch each time. Is that a helpful study tip? I hope so. Try to take advantage of it. You'll notice I do that. You wonder why I know this stuff so well? Because I've written it down 1,500 times, right? I know it because I've done it. That's the only reason I know it so well. And you guys can be here too if you just practice. All right. I want to manipulate this a little bit. But in order to manipulate this just a little bit more, I'm going to draw a gigantic synapse up here. And I'm going to teach you guys something. One more step, always more steps, because what do we need to do to ATP? If we want to bring that heart rate back down again, what do we need to do to cyclic AMP conversion? Increase, decrease, no change. If I bring it down, what's going to have to happen to the amount of cyclic AMP in the system? Increase, decrease, no change. Yeah, because an increase in cyclic AMP increases the heart rate. If I want to decrease the heart rate, I need to bring that cyclic AMP level back down. What about norepinephrine? If I want to decrease the heart rate, what do I need to do to the amount of norepinephrine in the system? Increase, decrease, no change. No. Yeah, right? It's when you think about it like that, using the flowchart, it's pretty logical. But students always miss that because they don't really think these systems as flowcharts. This must happen for this to happen. And this must happen for this to happen. And this must happen for this to happen, and so forth, so on in normal physiology. So if one piece of this puzzle breaks, say for instance, if I just magically have an increase in the amount of norepinephrine in the system, what's going to happen to that patient's heart rate? Increase, decrease, no change. What if that patient had more beta-1 adrenergic receptors? What if they magically had more beta-1 adrenergic receptors? What's going to happen to that patient's heart rate? Increase, decrease, no change. What if we were to somehow go in here and manipulate the amount of cyclic AMP in the system, and we gave them more cyclic AMP? What's going to happen to that patient's heart rate? Increase, decrease, no change. So this is what I love about flowcharts, is once you learn the normal flowchart, you can manipulate the crap out of this. And guess what? That's what drugs do. Whether it's pharmaceutical or recreational, those drugs are affecting this flowchart somehow. So for instance, you guys learned about a hormone that increases the number of beta-1 adrenergic beta adrenergic, or just all adrenergic, but primarily beta-1 adrenergic receptors, called an upregulation of beta-1 adrenergic receptors. And I'm not going to sit around and wait for you guys to come up with the answer, because most of you have probably forgotten, but it's thyroid hormone. So T3, T4, if we have more thyroid hormone in the system, there are more beta-1 adrenergic receptors in and on the autorhythmic cells. Correct, Tariq. You got it. If we decrease norepinephrine, it's before beta-1 adrenergic receptors. Now, that being said, you start decreasing the amount of norepinephrine in the system, you could potentially increase the number of beta-1 adrenergic receptors in the system to make them hypersensitive. If you have more beta-1 adrenergic receptors, they're more likely to be activated by less norepinephrine. But let's get back to here. If a patient has more T3, T4, they will have more beta-1 adrenergic receptors. So what would happen to that patient's heart rate? Increase, decrease, no change. And this is hyperthyroidism. That's why this is a major diagnostic tool for a hyperthyroid patient. They should have tachycardia because they have more beta-1 adrenergic receptors. Make sense? What about nicotine, right? Nicotine is upstream here. So I throw nicotine into this system. What's going to end up happening is that pre-post ganglionic neuron is going to be hyperactivated. And what's going to happen to the amount of norepinephrine being released? Increase, decrease, no change. If I throw nicotine into that system, tachycardia. Couple more, just two more, because it's important to know how things break down. Because as long as norepinephrine is being released via exocytosis, if we're releasing norepinephrine into the system, that norepinephrine will be binding to these beta-1 adrenergic receptors, and it will be activating them. As long as norepinephrine is in the system, it will activate beta-1 adrenergic receptors. Is that clear? So what we need to do to norepinephrine is get rid of it. Just like in AMP1, you guys learned about getting rid of acetylcholine at the neuromuscular junction. We have to get rid of norepinephrine at the synaptic junction. Gotsta. The way norepinephrine is taken care of is through a process of reuptake. So norepinephrine has a transporter found here in the synaptic Bouton membrane. This transporter helps move norepinephrine back into the terminal Bouton. That norepinephrine can sometimes be recycled, gets put into more vesicles, but often what will happen to it is it gets transported to the mitochondria. And there's lots of mitochondria found in these terminal Boutons. So mitochondria are almost always drawn like this crazy structure here. Okay. Now we'll get there in a second, but yes is the answer. Okay, these mitochondria have an enzyme found inside of them. Yay for enzymes. Called monoamine oxidase. Otherwise abbreviated as MAO. I call them MAO, but they aren't. Okay, MAO. MAO. They're normally just pronounced MAO, but I like MAO. MAO, MAO, MAO. What monoamine oxidase does is this breaks down norepinephrine and epinephrine. Okay, that's the function of monoamine oxidase, and we really only have that in the mitochondria of our cells. So norepinephrine and epinephrine don't degrade very fast in the bloodstream. They have to get taken back up into the cells of the body where there's mitochondria, and it breaks down the norepinephrine and epinephrine. So this is going to break it down, norepinephrine in this system. Okay, so there is a one down here. There was a norepinephrine reuptake transporter that helps to reuptake norepinephrine into the Bouton. And two, there is an enzyme called MAO found inside of the mitochondria that helps break down norepinephrine. So what would happen if we blocked the reuptake of norepinephrine? What's going to happen to the amount of norepinephrine in the synaptic cleft if we block this right here? We're blocking it. Increase, decrease, no change. What would happen to the amount of norepinephrine in the cleft? You guys, everybody that's done this so far has done great. Does it make sense? If we are no longer reuptaking it, more norepinephrine would be found inside that cleft. So what's going to happen? It's real easy from here, right? What do we have? We have this right here. We already drew this arrow. We just blocked the reuptake of norepinephrine. So what's going to happen to the norepinephrine in the system? Increase, decrease, no change. And therefore activates the beta-1 adrenergic receptors longer. Adenylate cyclase is going nuts. Cyclic AMP is going nuts. PKA is going nuts. You guys get the point, right? What's going to happen in that patient? Massive tachycardia. What's the name of this drug? Cocaine. This is what cocaine does. This is cocaine. Cocaine blocks the reuptake of norepinephrine. That's its function. That's its job. It has lots of other problems because of the amount of norepinephrine in the system. It just causes you to be basically on an adrenaline high all the time. If anybody's ever jumped out of a plane or done something crazy, got in a car accident, which isn't, typically on purpose, hopefully not, you get this adrenaline high, and that's supposedly what being on cocaine is like. It's just this permanent adrenaline high, which you crash pretty hard from afterwards. All right, what about MAO? What if we slow down the breakdown? What if we slow down the breakdown of norepinephrine? What's going to happen to the amount of norepinephrine in the system? Increase, decrease, no change. Yeah, and then it just gets recycled into vesicles. So these are called MAO inhibitors, and we give MAO inhibitors as an antidepressant. Okay, so that people that are depressed all the time, well, now they got a little bit more of a sympathetic response, a little bit more energy. So instead of the lethargy and stuff that's involved. So I just want to teach you guys that, one, flowcharts are freaking beautiful. I love them. I love the magic of changing colors to whatever the hell we want them to look like. But when you learn normal, you learn abnormal. Yeah, they definitely are more prone to crash. But you got to deal with side effects of lots of things. But once you know normal, you know abnormal. Does that make sense in terms of these flowcharts? Can you see that now? What if we have a mutation that slows down this funny channel? Or what if we have a mutation that makes it so that what if we had hyper or hypocalcemia? We know we're going to start to impact our heart rate somehow. Heart rate's pretty sensitive. It's sensitive, very sensitive. All right, we got to get going. We got to get rolling. But don't forget this flow chart, because remember, this is a beta-1 adrenergic receptor. And did we find beta-1 adrenergic receptors only on autorhythmic cells, or do we also find them on contractile myocardial cells? Oh, and if beta-1 adrenergic receptors are activated, guess what? Adenylate cyclase, ATP, cyclic AMP, PKA. Yeah, but not just heart rate increases. That's what I'm trying to tell you, right? If they're all over, beta-1 adrenergic receptors don't change their initial function. They're still using adenylate cyclase and cyclic AMP. So if we're going to be increasing stroke volume, if you've learned this, then you've learned this, right? You're not going to have to change your knowledge base. Now, there aren't funny channels or T-type transient channels in contractile myocardium, so this portion is going to have to change a little bit. Does that make sense? And let's just use, I love drawing things, but we're a little bit low on time. I have my YouTube videos and everything, so you guys can go into those YouTube videos to learn this as well. But let's just use our lecture for this. By the way, here's a cool little diagram. It kind of goes through, there you go, there's cocaine, nicotine. It doesn't talk about the MAO inhibitors, but you just did, caffeine, thyroid hormone. So all these different things that ultimately increase the amount of cyclic AMP, therefore PK, right? And that PK is going to go phosphorylate downstream things. So this is a cool picture to look through it will cover exactly what we just covered with our flow chart. Another great flow chart. I love, love, love flow charts. You know that. I got a cardiovascular control center. Found where? In the medulla oblongata. So here we got the CVCC. Cardioacceleratory, cardioinhibitory. Cardioacceleratory causes the release of norepinephrine onto beta-1 adrenergic receptors versus cardioinhibitory causes the release of acetylcholine onto muscarinic receptors. Now, if we're only impacting the heart rate, then we're the autorhythmic cells. And if you want to skip a few steps, this flowchart's great for it. Ultimately, muscarinic receptors are going to affect the funny channel, but they're also affecting the T-type calcium channel and this transient potassium channel. So we open up the transient potassium leak channel and we close down the funny. I'm going to just write this in because students, you guys, you know this, but you don't know this when it doesn't say it every single time. That's why I don't normally, whatever, it's out. So it's going to decrease sodium through the funny channel, and it's going to decrease calcium through the T-type calcium channel. And the combination of this both hyperpolarizes the cell and decreases the rate of depolarization. We did this quite a bit yesterday, so I hope you guys feel comfortable with that. Same thing over here. If we increase the sodium and calcium influx, we increase the rate of depolarization, therefore increasing the heart rate. You guys feel pretty comfortable with that? All right, let's move on to inotropes. So heart rate times stroke volume, heart rate times stroke volume. Heart rate, agents that affect the heart rate were called chronotropic. Agents or drugs that impact the stroke volume are called INO. INO means force. Inotropic agents, force agents. So they're going to change the force of the heart. Positive inotropes, negative inotropes. Well, what do you know? Positive inotrope, norepinephrine, epinephrine, T3, T4. Did you guys just learn about T3, T4? What's it going to cause an upregulation of? What is thyroid hormone going to cause an upregulation of? Yeah, it makes it so there's more beta-1 adrenergic receptors. And if there's more beta-1 adrenergic receptors, then there's more receptors for norepinephrine and epinephrine to bind to. Yesterday, we went into a lot of detail about digitalis, digitoxin, and how that is a cardioglycoside that blocks the sodium potassium ATP pump found on the heart. Just slows it down. And when we do that, it affects foxglove, you got it. When it does that, it impacts the NCX, the sodium calcium exchanger, and therefore more calcium builds up inside the cell. Negative inotropes. Potassium. We'll talk more about that, but not really. So basically, if there's just too much potassium in the system, it causes a prolongation of the action potential. I really don't want to get into it too much. Listen, too much potassium stops your heart. It's really complex because it has to do with the physics and kinetics of how potassium is small and can utilize calcium channels. And then when you start talking about calcium changes, then we're starting to talk about how calcium impacts sodium. And it's a very complex subject. So I'm not going to really get into it. You guys could memorize the fact that if we have too much potassium in the system, your heart's going to stop. It's going to stop beating if it's too much. So we got to be really careful with patients and their potassium levels. We are constantly monitoring our patients' potassium levels. Calcium channel blockers. I mean, if we block calcium channels, L-type calcium channels, does that sound like a good plan to block L-type calcium channels? Not normally. Not normally. But what if a patient needed to have their stroke volume come down for whatever reason? Then blocking those L-type calcium channels, not a bad idea. Remember, if we block these L-type calcium channels, then less calcium enters the cell. Does that make sense? And if there's less calcium entering the cell, then less ryanodine receptors get opened. Less ryanodine receptors, less calcium can leave. Decreases the amount of calcium versus normal, therefore the force of contraction starts to go down. So that's the idea there. Now here is how I have this all set up. And these terms will be important for the rest of the semester, not just today, not just this one small slide. These two terms are extremely important for you guys to learn. It's called intrinsic versus extrinsic controls. Now, they're not too hard to learn. They're not too hard to learn, but we got to learn them. Intrinsic tells us a local control. Intrinsic is within, it's local. It's in. Extrinsic tells you it's from without. Without? No, that's not right. It tells you that it's from outside. There we go. Intrinsic's from inside, extrinsic's from outside. And through the rest of the semester, you're going to be learning intrinsic and extrinsic controls. Intrinsic and extrinsic controls of your heart. Intrinsic and extrinsic controls of blood pressure. Intrinsic and extrinsic controls of respiration. Intrinsic and extrinsic controls of kidney function. Intrinsic and extrinsic controls of the digestive system. So please, you must understand these terms. Here's the easiest way to remember it. What two organs of our body communicate over a distance? Oh man, that was kind of my fault. I said it wrong. Yeah. So let's stop. Let me re-ask the question. What two organ systems of the body help with communication? Good. Yeah, that gets you guys on the right keywords. I apologize. Yeah, nervous system and endocrine. So if it's coming from outside of a local organ, doesn't it stand to reason that that must be related to the nervous system and the endocrine system potentially? One or the other or both. So extrinsic controls will always be either the nervous system, the endocrine system, or both, because they are extrinsic. They're coming from outside of that organ. Make sense? Awesome. The extrinsic control of stroke volume is all about the sympathetic nervous system. Okay, the CVCC is going to have a massive control on the extrinsic controls for stroke volume. What do you notice? Does it say parasympathetic? Why not? Why doesn't the parasympathetic nervous system have an extrinsic control? Poor parasympathetic. Why not though? What makes a target cell a target cell? So what is not found on contractile myocardium? Yeah. And what are those PNS receptors? Just to keep on this train, the same train. The A train? The B train? No. Parasympathetic, not A2, B2. No, for sure not. All adrenergic receptors are adrenaline, Heather. Just you'll get it. You went fast. That's all. That's all. I know how that works. Right? Muscarinic. So let's do this again, everybody. What are the only receptors for the parasympathetic nervous system found on a target organ? The answer is muscarinic. Keep working on that. Everybody needs to keep working on that, no matter what, because If you got to stop and slow yourself down and really think about it, then do that too. It really is important, because that's what we're going to be dealing with. Kidney system, respiratory system, digestive system, blood vessels. What if I told you blood vessels don't have muscarinic receptors? They only have adrenergic receptors. So what's the only nervous system that will have an extrinsic control on blood pressure? No muscarinic receptors, only adrenergic. Therefore, the only nervous system that can control blood pressure is sympathetic. Does that make sense? There's no receptor, then there's no function of that system. Awesome. Intrinsic controls come from within. This is going to be a little bit trickier for you guys to understand right now. Maybe, maybe not. We kind of have skipped a lecture, and we're going to talk about lecture on Monday, although I want to get directly into blood pressure, but we'll be fine. We'll use both. We'll use Monday for a little bit of everything. But intrinsic controls are coming from within the heart, from within the heart. And what I want you guys to think about is a pretty simple idea. If we have a chamber of the heart, I'm just going to draw, I'm going to draw a quick, relatively quick, oh shit, I'm just going to draw the imposter heart. It'll work. All right, and I'm going to divide this into our 4 chambers. And if I take and I fill these chambers, the right ventricle and left ventricle, with blood. Okay, I'm going to fill these up with blood. I don't know. Probably should have used a bluish color for this side. But bluish. It's blue blood. And then over on this side, I don't know. Where's my red? Here it is. We got oxygenated and poorly oxygenated. Whatever. Yeah, now it's black blood. It does the trick. It's okay. Now, if the heart beats, so just everybody watch. Stop trying to draw and follow along that way. Everybody just watch. There's a certain amount of blood in these chambers. And now the heart contracts, and it's going to create a certain amount of pressure. Depending on that amount of pressure, this blood is going to get shot out of the heart. Now, what if we did the exact same amount of pressure, but we add more blood to these chambers? What's going to happen to the amount of blood that can get ejected from the heart? If there's more blood available to begin with, what's going to happen to the amount of blood that can leave the heart? Increase, decrease, no change. Sweet. That was pretty easy to understand, wasn't it? Now, as the heart relaxes, remember this is called diastole. As that heart relaxes, this is called diastole. Well, the whole function of diastole, what's the function? If your heart's chilling and relaxing, what's going on that whole time as the heart relaxes? Yeah, you better believe the function of diastole is to refill the heart with blood. Oh yeah, oh yeah. So we got blood refilling the chambers of these hearts. So what's going to happen at the very end of diastole? Will we have a very high volume of blood relative to the end? No, the end of diastole, Tariq. So after we've refilled the heart with blood, it's going to have the highest volume of potential blood. Does that make sense? This is called end diastolic volume. So at the very end of the heart refilling with blood, there's a specific volume of blood inside of that heart. It's called end diastolic volume. We abbreviate that EDV. And it's going to be the maximum volume of blood in that heart. Maximum. maximum, I don't know how to spell maximum. I think that's right. Volume of, I'm just trying to go fast, blood in the heart. So what would happen if we increase the EDV? What's going to happen to the stroke volume? What's going to happen to the amount of blood that can leave the heart? So if we increase EDV, we increase stroke volume. What happens if we decrease EDV? Then we decrease stroke volume. Awesome. So what are some ways that we can increase that end diastolic volume? Well, what if you just drink more water? What's going to happen to your blood volume as you start drinking more and more water? Increase, decrease, no change. The water's got to go somewhere. So it goes from your stomach, your intestines, out into your blood, and then more blood is going to fill up these chambers, and your heart will beat stronger. Give it a try. Chug, not a gallon. Don't do that. Just chug a glass of water and then really pay attention to what your heart does. It'll be fast because your body's going to homeostase itself. That's a word, right? Homeostase. It's got to be the verb. There's another way. There's another way, and there's an easier way because you're not always drinking water. What is this other way? What big-ass blood vessel lies down here and what, a little bit smaller, even though the word makes it seem like it's bigger. Yeah, so we got the superior vena cava up here, and we got the inferior vena cava down here. Now, what I don't think students understand as they're first learning about the vena cava, because you guys just have a heart, and you guys, when you're first learning about it, you're basically just looking at a picture of, or a model of a heart, and you're not looking at the rest of the body. So let me show you guys how big the inferior vena cava is, in case you didn't look this up already. Okay, the inferior vena cava runs all the way from the base of the heart, and it runs all the way down to your pelvis. It's huge. It's absolutely ginormous. So don't let the word inferior trick you. Okay, it's not inferior in terms of size, it's just inferior in terms of direction. And all of the blood from the body, from kidneys, from liver, from spleen, all of that ends back into the inferior vena cava. It's big, it's real big. The second cool thing about veins in the inferior vena cava is that they are actually kind of stretchy. There's not a lot of muscle around them, but there is some muscle around it. This inferior vena cava right here has smooth muscle, just not bands of smooth muscle, but it does have smooth muscle that runs in the same direction as the inferior vena cava. These are called longitudinal smooth muscle. Don't worry too much about this yet. You're going to be learning more about this next week. But the idea here is if I take this inferior vena cava and I contract a muscle, what's going to happen to this inferior vena cava if I contract it? So now instead of vasodilation and vasoconstriction, what I do is I shorten it. That's what's so unique about having longitudinal muscles around a blood vessel. Normally they'd be circular, but in this case they're longitudinal. So now if I contract these muscles, it shortens the inferior vena cava. Yes, no, maybe so? Does that make sense? Now what if I told you that the inferior vena cava valves just like the heart does? So as I shorten it, it's going to prevent blood from flowing back this way. It cannot flow backwards. It can only flow forwards. So as I shorten the inferior vena cava, that's going to cause all of the blood that was down here in the inferior vena cava, it's going to cause it to go shooting into the heart. Does that make sense? It has nowhere else to go. So what's going to happen to the amount of blood returning to the heart if I have this? Good. This is called an increase in venous return. This is venous return. And it's primarily returning through the inferior vena cava because that's where most of your blood is at. In fact, the inferior vena cava is the #1 storage of blood in the body. It's a huge reservoir of blood. So the venous return, the IVC shortens, and what that does is it's going to increase the venous return into the heart. And if I increase the venous return, what's going to happen to the end diastolic volume? Increase, decrease, no change. More blood's entering the heart. Therefore, the end diastolic volume would also increase. Does that make sense? And as long as I increase that EDV, what's going to happen to that stroke volume? Increase, decrease, no change. Dun, 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 which is exactly what's going on here. We have an intrinsic control. How much venous return is returning to that heart? The venous return has to do with the EDV, and as EDV changes, it changes the stroke volume. More blood in, more blood out. It does, Heather, but to a much lesser degree. And if you actually look at pictures of the superior vena cava, it's really, really here. This one will work. Look, there's that superior vena cava. That's it. Just that little tiny portion right there. So it's not, and think gravity, right? This isn't a reservoir. It's not really holding blood because blood's just going to dump down into the right atrium via gravity alone. So it's not storing it away. So shortening it's not going to do a lot of good. Make sense? This, we got to have blood pressure to push it back up against gravity into the heart. So this inferior vena cava is a really powerful reservoir. Question. Yeah, imagine, Tariq, somebody that just got in a car accident. And now imagine that somebody that just got in a car accident has internal bleeding. And now imagine that that person that just got in a car accident has internal bleeding from the inferior vena cava. It's not much you're going to do. So it's one of the places you hope never gets injured because it's very, very difficult to stop the bleeding. Stroke volume. It is. It's in the center. It's kind of being protected by the spinal cord. But yeah. All right. Stroke volume. Stroke volume. We just learned there are intrinsic and extrinsic controls. The primary way we can increase stroke volume is through the sympathetic nervous system. And if you have sympathetic activity, you guys are going to learn here in just a minute how that is going to increase the force that that heart can contract. At the same time, what did I just teach you a couple minutes ago? What are the only types of receptors found on blood vessels? Adrenergic or muscarinic? No, don't say beta 1, Tariq. Please don't. Please don't. That's the confusing part of the autonomic nervous system. But beta 1's only found on the heart. Beta 1's only found on the heart. We got to be really general, and then we hone ourselves in. Yeah. So blood vessels. Yeah, you got it. 1 second. I am talking about blood vessels in my question. So everybody stop, think about blood vessels, not the heart. What's the only type of receptor found on blood vessels? Adrenergic or muscarinic? Good, adrenergic. And if you go back, is that clear to everybody that we have only adrenergic? And we've actually done this. We did this in our review, whenever this was, yesterday, day before, I don't know, sympathetic nervous system. When we were going through the different adrenergic receptors, we said almost all smooth muscles and most of the blood vessels of the body have alpha-1 adrenergic, whereas coronary blood vessels and skeletal muscle blood vessels have beta-2. So when you're dealing with blood vessels, there are two answers that it can be, alpha-1, beta-2. What's the difference between those two and their function? Alpha-1 is excitatory and beta-2 is inhibitory. Which of these, excitatory or inhibitory, is going to cause the contraction of the inferior vena cava? So let's put that all together. What type of adrenergic receptors are you going to find on the inferior vena cava? Does that make sense, y'all? No, not beta-2, Tariq, not beta-2. Beta-2, you're not going to have excitatory and inhibitory. You're only going to have excitatory. And what do we do if we want to cause the relaxation of that particular blood vessel? Shit, just get rid of the sympathetic nervous system. If the sympathetic nervous system's gone, then we're not activating alpha-1 and it's going to relax. Okay? We want a really fine regulation of our blood vessels. It's auto. What do you mean by auto? Help me understand your question there. It is because, remember, the parasympathetic nervous system and the sympathetic nervous system are a teeter-totter. So as parasympathetic nervous system increases, what's going to happen to the sympathetic nervous system? It will decrease. And just by decreasing the sympathetic nervous system, we slow down the release of norepinephrine, and there we slow down the activation of the alpha-1 adrenergic receptor. Does that make sense? It is. It is very smart. It's got very, very fine-tuned control of everything. Okay, cool. So, extrinsic controls, sympathetic stimulation. All right. Well, we're going to come back to Frank Starling law of the heart. It's all about preload. Preload is how much blood is entering the heart to begin with. But as we just mentioned in our little simple drawing, what do we know? If I have more preload, that means there's more blood in. So what's going to happen to the amount of blood that can be ejected out? Increase, decrease, no change. So the short and sweet of Frank Starling law is more blood that enters the heart, the more blood can leave the heart. And it has to do with actin-myosin overlap stuff, and I'll teach you all about that on Monday. But more, can you guys just write this down then? This is actually the easiest way to remember Frank Starling law of the heart. So write Frank Starling law of the heart, and you could be like summarized in your notes. We're going to define it. More blood in, more blood out. It's really that simple when it comes down to it. Frank Starling law of the heart. As more blood enters the heart, more blood will leave the heart. And story, unless there's heart failure. But in normal physiology, more blood in, more blood out. Frank Starling law of the heart. Did everybody get that written down for Frank Starling law of the heart? More blood in, more blood out? Anybody need more time? Just let me know if you need more time. Because the last thing I'm going to cover today is complicated. And I'm going to actually give you guys this as an assignment. Dr. H, why would you do this to us? Because I can. So this assignment that you guys are going to do, because summer, It's always the excuse now. You guys can, instead of having those because physiology memes that are out there, you could be like, because summer physiology. There you go. You're welcome. No, I just think it's important to think our way through problem sets. But this isn't as hard as it seems at first. There's a lot going on in this flowchart, right? There's a lot of arrows. But what I want you guys to think about is this diagram that we've already covered. I know when you look at it, there's a lot going on here too, but I have flowcharts for everything, and ultimately what we're looking at is this little thing right here for relaxation, and this little thing right here for contraction. And what we said was that if we have excitation-contraction coupling, that an autorhythmic cell sends an action potential to the contractile myocardium. When it makes it to the contractile myocardium, it's going to activate the L-type calcium channels, and calcium is going to start to move into this cell, thus activating the calcium-sensitive calcium channels, ryanodine receptors, opening them, at which time the big-ass bag of calcium can release that calcium into the ICF. What's going to happen to the concentration of calcium in the ICF? It's increasing. And what do we know about the force of contraction and calcium? If there's an increase in calcium, what's going to happen to the force of contraction? Increase, decrease, no change. Yes, Heather. But there's always going to probably, oh, that's a great answer, huh? I can't imagine a time when there wouldn't be enough calcium. I would imagine that as long as L-type calcium channels, unless you're using a complete L-type calcium channel blocker, it would have to be a really severe inhibitory drug and it would kill your patient. You're never going to stop the opening RYRs. But if you're thinking maybe like a cardiotoxin, there might be one out there. I don't know. That's not one that I've looked into, but I wouldn't doubt it, sadly enough. But back on target here. We just said that as calcium increases, the force of contraction increases. What do we know about the sympathetic nervous system? What's that going to do to the strength that your heart beats? Increase, decrease, no change. So let's tie those two logical trains of thoughts together. What do you think the sympathetic nervous system does to the amount of calcium in the ICF? Increase, decrease, no change. And that's exactly how the sympathetic nervous system is going to function. Sympathetic nervous system is going to make it so there's more calcium. It's going to hold those L-type calcium channels open just a little bit longer. It's going to hold the L-type calcium channels open just a little bit longer. When would the L-type calcium channels normally close? When the action potential stops, right? True story? But what if the action potential stops, but these L-type calcium channels open just a little bit longer? Then they close. But I'm talking, a couple, 100 milliseconds or something. But it's enough time in the human body for that to take an effect. If I hold these L-type calcium channels open just a little bit longer, more calcium can enter the cell. Make sense? And again, I did this before, but what if I sped up this ATP pump? What if I sped up the calcium pump? What's going to happen to the rate calcium is leaving the ICF? Increase, decrease, no change. And so intuitively, you guys might start to think that there's going to be less calcium here. But what do we know about the movement of calcium back out the ryanodine receptor is that it's reliant on a concentration gradient. So what's going to happen to the concentration of calcium inside the SR if I speed this pump up? Increase, decrease, no change. And therefore, when the ryanodine receptors are open, what's going to happen to the rate that calcium can move back out? Increase, decrease, no change. And therefore, calcium binds to the faster. Does that make sense? As long as the ryanodine receptors are open, but then they close, and the pump's still moving faster, so what's going to happen to the rate that we can remove calcium? What's going to happen to the rate that we can remove calcium from the ICF as that heart is relaxing? Is it going to go faster or is it going to go slower? Remember, this pump's still working faster in this particular example that I'm giving right now. So what's going to happen to the rate? Increase, decrease, no change. That we remove calcium. Yeah, exactly. As long as the ryanodine receptors are open, calcium's still moving faster, but it can come back out and bind to troponin faster. Now, when the ryanodine receptors close, calcium's removed from the ICF faster, thus we relax faster. Everything's going faster. Because what have I been trying to teach you? It doesn't matter how fast your heart beats. It doesn't matter how strong your heart beats. If there's no damn blood in a heart, who cares? The heart must refill with blood. The heart must refill with blood. And in order for that heart to refill with blood, as it's beating faster, well, it better be relaxing faster too. Does that make sense? It can't just contract faster. It must relax faster. Exactly, Heather. The faster it relaxes, the faster it refills. Does that make sense to everybody? And that's why 190, you're starting to get to a point where the heart can't refill with blood properly. You get up to 270, 280, now it's basically not refilling with blood. It's going too fast. It's pumping that blood out faster than it could refill. So that's why we try to keep our heart rates under 180. It tends to be Goldilocks. Okay, so what is your guys' assignment? Is I want everybody not to draw this flow chart, but to understand this flow chart. In order to understand this flow chart right here, you're going to end up drawing a diagram. You're going to draw this diagram right here of a cardiomyocyte. You can draw in some of the normal function, get rid of all of these excess flow charts and stuff. And what I want you to do is I want you to fill in what's happening. Okay, so we're going to have beta-1 adrenergic receptors, norepinephrine, epinephrine, and I want you to draw those into this diagram. Do you think I'm making you do this blind? Or is there potentially a place for you to go and watch me do that somewhere? You guys already know the answer. Would I ever make you guys do this completely blind? I don't think, that's not my style. So what do you got? You have all of these YouTube videos. Check it out. Intrinsic and extrinsic controls of myocardial cells right here. We've just walked through this. You should have a basic idea of how this works already. Well, there you go. There's your diagram of that cardiomyocyte. I get you guys started off. It's a 38-minute lecture, but by the time it's all done, you will have drawn in beta-1 adrenergic receptors, GPCRs, G-stimulatory, right? Going and exciting things. Look, adenylate cyclase, cyclic AMP, PKA. So not brand new, right? Nothing here should be brand new. Now we're just taking these ideas and we're tying them all together. Does that make sense? I'm not trying I'm really not trying to make you guys open up a black box of an, enigma that just makes you guys want to quit. I'm trying to give you guys all the pieces of the puzzle. There's NCX right there. All the things that you guys have already learned, and now we got one big thing that pulls it all together. You guys understand what you need to do? You need to draw out one of these and put in what's going on with beta-1 adrenergic receptors. Easy peasy. Then you just text me that picture. So draw it in your notes and text me the picture. Easy? You guys got it? We're not submitting on Blackboard, nothing. And I'll look it over and I'll say, awesome job, or maybe you want to add this, maybe you want to add that. 2 thumbs up. Lovely. Okay, well, next week, oh, it's just due by next lab. That's fine. By next lecture, by Monday. But the sooner the better. Fresh on your mind, in my opinion. But not everybody has that time today, so do it when you can. All right, so next week, next Monday, we're going to finalize our cardiophysiology, move on to blood pressure. And don't forget, your exam is already next weekend. I will have everything finished grading. I started last night. People are doing pretty good. And I will get all those grades submitted probably by, I don't know, tomorrow. Yeah, tomorrow at the latest. And I'll make sure all the rest of your grades are up to date by tomorrow as well, so you guys know where you're at. All right, cool. Please text me if you have any questions. You guys have been doing a great job with that. Study, study, study. Y'all too, have a great weekend. Study and play. Not just study, study, study. I forgot to say that. Study and play. Got to balance.